Surface biometrics operate with a measurable margin of error. Commercial facial recognition algorithms currently accept the wrong user roughly 2% of the time, but fingerprint scanners perform better, still clearing an incorrect match in 1 out of every 1,000 attempts. This logarithmic chart compares those error margins against the human retina. Retinal scanning operates at a false acceptance rate of less than 0.0001%, 1 in a million. Statistically, it is the most rigorous human identifier currently mapped. The architecture of the retinal vascular system is highly chaotic. Even monozygotic identical twins develop structurally distinct arterial layouts. Furthermore, because this network is encased deep within the globe of the eye, it cannot be inadvertently left on a glass surface or captured from a distance by a standard high-resolution camera. The security of this modality stems directly from its anatomical isolation. It relies on protected internal neural tissue rather than exposed environmental surfaces. The retina is a multi-layered neural tissue lining the posterior wall of the eye. It sits at the absolute rear of the ocular chamber, converting incident light into electrical signals. The vascular tree driving this tissue originates from a single biological and computational root, the optic disc. Here, the central retinal artery and vein breach the interior, immediately branching outward across the inner strata. Near the center lies the fovea and macula, an avascular zone densely packed with cone photoreceptors for high-acuity vision. Any biometric mapping system must navigate precisely around this empty void. This intricate network of vessels is established entirely during fetal development. Under healthy biological conditions, the geometric structure remains strictly stable for a human's entire lifespan. Engineering this system requires translating a buried, microscopic biological pattern into a highly compressed 1024-byte mathematical template. Standard clinical acquisition relies on the fundus camera. These systems pulse green light at wavelengths between 550 and 640 nanometers, specifically because hemoglobin heavily absorbs it, maximizing the optical contrast between the vessels and the surrounding tissue. However, a fundus camera only records a flat, two-dimensional projection. This lack of depth leaves the system theoretically vulnerable to physical spoofing. Optical coherence tomography, or OCT, solves this by projecting 840-nanometer near-infrared light. This penetrates the tissue, capturing true volumetric, three-dimensional cross-sections of the interior. OCT data reveals the vascular network is not a flat web, but physically separated into depth zones. The OCTA nomenclature classifies these into a superficial capillary plexus in the inner layers, distinct from the deep capillary plexus below. Mapping depth creates an intrinsic mechanism for liveness detection. Accurately replicating separate, flowing microcapillary networks across distinct biological strata makes three-dimensional spoofing mathematically near impossible. But whether acquiring a 2D projection or a 3D volume, the immediate output is just raw, heavily distorted pixel data. The system must algorithmically purify the signal before any true extraction can occur. The first step in signal purification is isolating the green channel from the raw RGB image array. This guarantees the highest signal-to-noise ratio, yielding a standard deviation in vessel contrast of roughly 0.47. Because the eye is a spherical chamber, the resulting images suffer from severe, non-uniform illumination. This histogram illustrates the application of contrast-limited adaptive histogram equalization, or CLH. It computationally flattens the light gradient across the entire array. Historically, isolating the actual vessels from the background tissue relied on matched Gaussian filters, rotated across multiple axes. But these early computer vision techniques stalled at around 71% sensitivity. The modern standard abandons simple filters for deep learning, specifically relying on encoder-decoder convolutional neural networks, known as UNets. By pushing the image through multiple downsampling and upsampling paths, the UNet boosts extraction sensitivity to 82% and specificity to over 98%. The output of this neural network is a perfectly isolated binary vessel map, completely stripped of the fovea, the optic disc glow, and all extraneous cellular tissue. Yet, a grid of binary pixels cannot be efficiently queried against a database of millions. The biometric pipeline must now translate image processing into abstract topological mathematics. To do this, the system maps the binary pixel array as a directed acyclic graph. The physical vessels become mathematical edges, and their intersections become connecting nodes. The algorithm localizes the central optic disc, establishing it as the absolute origin coordinate, 0, 0. The most critical topological features are the bifurcations. At every point where a parent vessel splits into two daughters, the system plots a degree 3 node, recording its precise x and y location, as well as the branching angle, theta. These branching angles are highly predictable. They are strictly governed by Murray's law, a physiological principle dictating that vessels will bifurcate at precise geometric angles to minimize the energetic cost of pumping blood. Beyond individual nodes, the algorithm calculates the macro statistics of the entire web. By applying a box counting method to the vessel skeleton, it extracts the network's overall fractal dimension. A healthy, properly segmented retinal graph will consistently yield a fractal dimension of approximately 1.70. With these variables calculated, the chaotic biology of the eye is compressed into a stable, immutable feature vector, a secure template ready for database enrollment and query matching. When a user attempts to authenticate, a live query graph is generated through the entire pipeline. The system must then compare this input against the secure database. This formula plots Euclidean distance, computing the variance between multi-dimensional vectors by measuring direct mathematical differences. But sophisticated pipelines use graph edit distance, calculating the minimum cost to morph a live network topology until it matches the stored template. The resulting difference score is checked against a strict threshold, tau. If the deviation falls below that threshold, the system grants access, executing this entire decision sequence in under 2 seconds. While this yields an extraordinary 98.5% recognition accuracy, the framework still has vulnerabilities. High-resolution 3D spoofing remains a concern, but the system's long-term stability is more frequently compromised by the user's own physiological changes. The human systemic state is rarely static. Biological decay poses the ultimate challenge to biometric security. This phenomenon is known as template aging. Over several years, a user's active biological signature can drift away from their enrolled baseline template, eventually causing the system to reject the genuine user. The geometric properties used to verify identity, such as the tortuosity index and the arteriovenous ratio, serve as dynamic biomarkers for pathology. A patient developing diabetic retinopathy will structurally alter their own graph. The disease spawns microaneurysms and forces erratic neovascularization, abruptly shifting the coordinates of previously stable bifurcations. Hypertensive retinopathy induces pressure changes that warp the diameter ratio between arteries and veins. This eventually causes vessels to physically cross over and crush one another at specific nodes, a process called AV nicking. Emerging clinical research demonstrates that these alterations often occur before major symptoms. Microscopic shifts in vessel tortuosity, or curvature, strongly correlate with brain amyloid load, acting as an early biomarker for Alzheimer's disease. The current imperative in machine learning is to engineer algorithms capable of decoupling these traits, systems that can isolate invariant topological identity markers while accurately tracking disease-induced physiological changes. The retinal vascular pattern integrates cryptographic security with a clinical diagnostic overview of the human systemic state.